Good evening, everybody. First of all, I would like to thank the scientific committee for the invitation. It's really a pleasure for me to participate at this annual scientific day. My presentation today will focus on the immune escape mechanism adopted by leukemic cells to evade immune recognition of donor T cells after hematopoietic stem cell transplantation. As you already know, the best therapeutic option for most patients suffering of acute myeloid leukemia is allogeneic hematopoietic stem cell transplantation, combining high-dose chemotherapy with cancer immunotherapy. Donor T cells, in fact present as part of the graft, are able to recognize on leukemic cells tumor antigens and minor and major histocompatibility antigens mismatched between the patient and the donor, exerting an anti-leukemic activity or the so-called graft versus leukemia effect. Still, relapse after transplantation remains an open issue for many patients, irrespectively from the type of transplant, and little is known about its biology. The main focus of our lab is to study the biology of acute myeloid leukemia relapse after transplantation. It is almost consolidated that leukemia clonal evolution could be explained in an evolutionary perspective. Our work hypothesis is that the changes documented at relapse in the disease clonal structure and immunogenicity may represent the end results of a process of selection, characterized by the disappearance of the most immunogenic clones and the outgrowth of variants characterized by genomic and epigenetic features that confers immune resistance to the therapeutic control of transplantation. A peculiar example of this paradigm was the observation made by our group in the context of the haploidentical transplantation setting. In this context, the mismatched HLA molecules represent the main targets of the graft versus leukemia response. We observe that leukemic cells at post-transplantation relapse are frequently characterized by the genomic loss of the mismatched HLA haplotype. This is not due by a simple deletion, but through a mechanism that leads to a copy-neutral loss of heterozygosity. This genomic alteration is stable and irreversible, involves both HLA class I and HLA class II molecules, and does not affect the overall surface expression of HLA class II molecules. By this mechanism, in vitro donor T cells lose their ability to recognize and proliferate specifically in response to post-transplantation leukemic blasts, but retain the ability to recognize blasts collected at diagnosis. Thus, by this mechanism, HLA-locked leukemic variants are possibly able to evade the donor immune system. We are now conducting an international multicentric study to evaluate the incidence of this alteration in the different transplantation settings. As you can see from the pie chart, this genomic alteration is more prevalent in the haploidentical cohort, but could be observed with a lower incidence also in the other allogeneic transplantation settings. However, this alteration does not explain all the post-transplantation relapses observed, so we next investigated whether immune escape could also explain the other cases of relapse after allogeneic transplantation. We performed a systematic comparison between purified AML blasts collected at the time of diagnosis and at relapse after transplantation, and as control at relapse after solely chemotherapy before transplantation. High-throughput technologies and immune phenotypic analysis were applied initially on an internal discovery cohort and then validated on an international and independent series of patients. Interesting results were obtained by the expression data performed on the discovery cohort that revealed that the top deregulated biological processes between leukemia cells collected at diagnosis and relapse after transplantation were immune-related, and here represented by the red bars in the chart. And by a semantic similarity analysis, we were able to group the gene ontology terms identified in the previous analysis into two main biological clusters, one related to T-cell co-stimulation and the other to antigen processing and presentation via HLA class II molecules. Firstly, I show you the validation results regarding the co-stimulatory asset. Paired leukemic samples of 33 different patients were assessed by flow cytometry. As you can appreciate, at relapse there is a consistent increase in the expression of coinhibitory molecules, which gets the significance for PD-L1, B7-H3, and PVRL2, and the significant downregulation of molecules involved in the T-cell activation, like CD11A. On the same leukemic samples, we studied the expression on the T cells of the cognate receptors of the ligands previously assessed. We observed a general deregulation of all these molecules on T cells from leukemia samples compared to the ones from healthy controls. And in particular, we evidenced a marked expression of PD-1 receptor on post-transplantation T cells, and the loss of the CD28 expression. Taken together, this data demonstrated that the co-stimulatory interface between T cells and leukemia changes significantly after allogeneic transplantation, with the loss of co-stimulatory interactions like CD28, CD80, and ICAM1, CD11A, and the enforcement of inhibitory ones, like the PD-1, PD-L1 asset. Now I'm showing you the results on antigen presentation. We and the group of DiPersio observed at the mRNA levels a significant downregulation in post-transplant leukemia cells of HLA class II genes and of their master regulator C2TA. This downregulation was then confirmed and validated on an extended cohort by immunophenotypic analysis that assess the surface protein expression. This analysis reveals also that HLA class I molecules remain mainly unaffected. We next confirmed our results on an independent validation cohort. We performed a clustering analysis on the changes in the surface expression between leukemic cells at diagnosis and at relapse after transplantation of all the molecules previously assessed. In both the discovery and validation cohorts, these analyses clearly separate the patient with a downregulation of HLA class II molecules from the ones characterized by the deregulation of coinhibitory molecules, suggesting that these two mechanisms are largely non-overlapping. Moreover, in both the cohorts, these two mechanisms are not associated to a specific type of transplant. In fact, matched and mismatched transplants are shuffled among the two relapse modalities. Altogether, these two mechanisms account for up to the 50% of relapses after transplantation. Next, we were interested in the therapeutic implications of these two relapse modalities. The upregulation of inhibitory ligands involves interferon-related genes and is worsened by inflammation. This inhibitory asset can be blocked by immune checkpoint molecules. In fact, in vitro, donor T cells respond better to post-transplant leukemia cells in the presence of an anti-PD-L1 monoclonal antibody compared to the non-treated controls. Also, HLA class II molecules are genes sensitive to inflammatory signals, and their expression on leukemia cells could be recovered upon exposure to interferon gamma. In co-culture experiments, donor-derived T cells collected from a patient who experienced a relapse with a loss of HLA class II expression respond to the leukemia at diagnosis, and not to its relapse counterpart. Interestingly, this recognition could be restored upon the pre-exposure of leukemia cells to interferon gamma. Now, in our lab, we are focusing our attention on the HLA class II downregulation to better define its molecular mechanisms. As I showed before, for at least some patients, the promoter of C2TA, that is the master regulator of all HLA class II molecules, is downregulated, and possibly this could be due for the hypermethylation of its promoter. Data from our lab show that the chromatin repressor complex 2, PRC2, could be involved in the HLA class II downregulation, and that inhibiting some components of its catalytic domain, like EZH2 or EED, could restore HLA class II expression with functional consequence. But how we demonstrated this? Firstly, in order to have a sufficient amount of cells for all the analyses, we generated patient-derived xenograft models from leukemia patients with a loss of HLA class II expression at transplantation relapse. We confirmed that after engraftment, the leukemia phenotype was maintained. And then we analyzed the gene expression profile, the chromatin accessibility, and the genome-wide methylation profiles. The results of these three techniques were integrated with the MOFA analysis that led us to identify that the most affected gene was EZH2. So next, by looking at ATAC-seq peaks, we confirmed a more compact chromatin in the promoter of HLA class II and C2TA genes in leukemia blasts collected at relapse compared to the ones at diagnosis. Interestingly, the expression of HLA class II molecules could be reinduced and increased in the presence of EZH2 inhibitors. This upregulation of HLA class II molecules has a functional impact, since in co-culture experiments, CD4 T cells are less able to be activated or to proliferate in the presence of leukemia blasts characterized by a low absent expression of HLA class II molecules. Both CD4 activation and proliferation increased when leukemia blasts were pre-exposed to EZH2 inhibitors, to levels similar to the interferon gamma treatment. So I've shown you how genomic and epigenetic mechanisms confer the ability to leukemia cells to evade the donor immune system after transplantation, thus leading to relapse. Each mechanism has a different incidence in the different transplantation settings. All these findings address the relevance of studying relapse biology, since based on each of the identified immune evasion mechanisms, specific therapeutic intervention could be implemented or designed, like for example the retransplantation from a different donor or the use of bispecific antibodies for the genomic HLA loss variants, or the induction of a mild inflammatory state, or the usage of epigenetic drugs for the downregulation of HLA class II molecules, or immune checkpoint therapies for the latter mechanism identified. In conclusion, from my talk, I hope I have convinced you that relapse biology should inform precision medicine approaches, fulfilling the idea and the logic of personalized therapies. Still, there is a proportion of post-transplantation diseases for which we do not know the mechanism at the base of relapse. To date, we revealed an impaired leukemia recognition by the loss or deregulation of the presentation machinery and the deregulation of immune checkpoint ligands. But there are other strategies by which leukemia cells could evade from the post-transplant immune system that should and could be investigated, as summarized in this cartoon. Finally, I want to thank all the people that contributed to this work, all my group headed by Luca Vago, Fabio Ciceri, and the bone marrow transplantation unit of our institute, and all our internal and external collaborators located inside and outside Europe, and all of you for attention.